Thanks, everyone, and thanks, Jon, for that talk. I think we'll have a little bit of overlap, actually, because we like to call ourselves chemical language modeling people. So it's actually a lot of similar ideas. So I work in the molecular AI department at AstraZeneca, and I'll get into what we do exactly, but first I'll just say a little bit about AstraZeneca. I won't spend very long on this except to point out we're a fairly large company, so our revenue is around nearly $40 billion per year, and the revenue for R&D is around $10 billion per year. That makes us around the 13th largest pharma company. So while these are big numbers, they're not actually enormously big for the pharmaceutical industry. We have some values stuff about the objectives of AstraZeneca as a company, and I won't get into them in detail except to say that we very consciously do put sort of science and innovation at the core of our activities, and I hope I can motivate you that it's not quite just corporate kind of doublespeak, and that we do actually try and deliver on that and actually all of these pieces. If we look at us from a global perspective, we have three R&D centers. So these are where we do actually development. One of them is in Gothenburg in Mölndal, actually, and that's where I'm located. Our other one is in Cambridge in the UK, and then in Gaithersburg in the US. And that's really where we do a lot of the development and scientific effort. Our other site in Sweden is in Södertälje, close to Stockholm, and that's mainly manufacturing. So roughly where we are, in particular, our presence in Sweden. So we have around 7,600 employees in Sweden, and those are split, as I say, between these two sites, and we contribute some amount to the Swedish revenue by paying our taxes. All right, so AstraZeneca does the full stack of sort of pharmaceutical activities, from really very early stage genomics, target identification work, all the way through to sort of manufacturing and clinical delivery and monitoring. We can split this enormous process into, sort of the late or clinical piece, and then the sort of early discovery piece. And, compute actually is used everywhere along this whole stack. But I work particularly in drug design, in early discovery. And in particular, I work in a department called molecular AI. Ola is our boss, and he was actually invited here. So I'm here, wearing his mask. He was unfortunately unable to make it, and I'm going to show you a little bit about what we do. And these are fairly generic deck of slides, but I'll try and reflect on our compute sort of aspects of it, especially. To sort of contextualize what happens in early stage discovery, what happens before the drug discovery process is a target is identified. So this is usually a protein, or we identify some particular disease-causing phenotype. So we have to have a target in place, and that can be done through genomics. And then our job, where I sit, is basically to go from a starting point molecule, which might be weakly active against this target, and convert it into a candidate drug that's suitable for ultimately evaluation in the clinic. And that has to improve its potency and also make sure it has no side effects, the safety profile. It must also be able to be absorbed and be metabolically stable, and all these things. And we essentially operate in an iterative cycle, which we call the design, make, test, analyze cycle, the DMTA cycle. And this is a repeated process that we do in order to improve molecules. So it's like an iterative molecular optimization problem. And this we solve in the lab, but we also are increasingly solving it using everyone's favorite digital twins. So in particular, when it comes to trying to accelerate this process using digital technology, where I sit, and what I'll talk about today is really methods for accelerating the design of molecules, and also a little bit about methods for accelerating the synthesis of molecules. But there's also lots of exciting stuff happening around, like synthesis robotics for accelerating the testing and analysis of data, which I won't talk about at all. So we've heard about language models already. It's very convenient because that's exactly what we do. So this is a picture of a molecule. And you can actually, so a molecule is made up of atoms joined together. And you can represent this as text. So here, each atom corresponds to one letter. And there's some specific grammar around these representations that dictate how these atoms are connected. But this is very analogous to letters and words in language modeling. And so because of that, we can basically steal all of the cool technology that's done in the natural language processing technology in order to basically create chemical language models. So our initial models were all kind of sequence recurrent neural networks, and we also have transformers now, which I'll talk about in a slide or two. I think actually we kind of covered this already, but just to sort of make it very explicit, here is a small character set. These colors represent the probability distribution over that character set encoded in a neural network. So you can see the darker the color, the more likely the symbol is. And these models go token by token. So we sample from this distribution, maybe we pick oxygen, and then we put an oxygen in our picture, and then we kind of go step by step through this whole process, and each time we kind of select a new character to add, and as we go, you can see the molecule is being drawn over here. And then at the end, we emit a special end token to indicate that the molecule is complete. And that's the core of these generative language models that we use for chemistry. So you can then take this kind of system and you can build cool machinery out of it. And the machinery that we build in my team is called the REINVENT ecosystem, and it is a generative reinforcement learning approach based on these kind of technologies. So we have the generative model that you just saw that emits new molecules, new words, and then we pair that with an in silico assessment where we kind of rate how good these molecules are, and we pass that feedback to the generative model using reinforcement learning. And then we can kind of iteratively steer the generated molecules to produce the kind of molecules that we are interested in. This is a kind of different paradigm for computational chemistry in the pharma industry. Traditionally, the sort of virtual screening type approach involved making a big list of potential molecules, and we do this also at AstraZeneca, and this can be at the billion molecule scale, and then searching through that virtual library for interesting molecules. But using this kind of iterative approach, we have a probabilistic encoding of the rules of chemistry. And then these models can describe theoretically really enormous numbers or really enormous variety of chemistry. But by guiding them in this way, the number of sort of oracle evaluations, the number of calls that we make to the scoring function is usually much lower. And that lets us use kind of more complicated scoring functions as well. So hopefully my video is working. This is a picture of a protein here, this big structure. And here you can see our REINVENT agent starting from nothing, inventing molecules that fit into this binding cavity in the protein. And the y-axis here is our docking score. The more negative it is, the better we're fitting into that cavity. So you can see in a few hundred evaluations, we're able to generate, a good set. Maybe you say -11, we would call those hits in virtual screening. We can generate a very large number of ideas, starting points for chemistry really quickly using these models. So this is much quicker than it would take to screen a big database at this level of theory. Okay, so I want to just show you, one other little piece, and this slide is really to emphasize that these are ruthlessly efficient optimizers. So I took our interface. By the way, we make a web interface available internally to our users, so they communicate via a web app, and we actually spawn these jobs on our usually internal HPC or on the cloud as a backend. But I opened up our web app, and just for fun, I said, give me a molecule with as many rings as possible. So this is like a not very realistic question, right? And you can see we start off with something that looks reasonable, but I've told it to give me more rings. So we go through this phase where we're starting to get unreasonable. And then we get to this thing over here, which looks, I don't know, like satellite solar panels, maybe. Nothing like a molecule. I mean, it's a syntactically valid molecule, but it's very far from the prior, from where this model started. And then at that phase, I invert the scoring function. So I say, suddenly give me no rings. And even after the agent has been traveling for 800 epochs with the scoring function, it can immediately deal with the changing conditions and produce entirely linear aliphatic molecules. So the point behind this demonstration, not that any of these are kind of relevant molecules, but the point is that if we can define an objective function for our reinforcement learning agents, they can solve the problem. So the name of the game becomes constructing accurate and meaningful scoring functions that we can use to assess these molecules, because the optimizers are of sufficient quality to solve the problems that we propose. And quite quickly, you can see they take very few evaluations to work out what needs to happen. Okay, so we started with the REINVENT project really in production in end of 2019, beginning of 2020. That's actually when I joined Astra. It was just being launched, and we had this kind of atom by atom construction that I've shown you. But since then, we've grown a lot in terms of other tools, solving different kinds of problems. So expanding upon existing scaffolds, we have linking tools. And then we also have transformers now, which we've just kind of productionized this year, and I'll talk a little bit more about them in their own slide. But obviously, we have a lot more things in the pipeline. What I think is nice about this, and going back to our kind of value statement, is actually everything is open source. So all of these models, all the architectures, everything are on GitHub. You can download them and use them yourself. So I think the transformers are yet to be pushed, but when the paper is published, they will be available. And we also publish about this ecosystem very widely. So this is just kind of like a humble brag of scientific publications that our group has generated in the last 2 or 3 years around this material. But it's done partially because we do open science, but also because of this model lets us collaborate very broadly. So we have collaborators here with Max Planck Institutes. We work with the team at Aalto University. We have lots of collaborations with Chalmers. And some of this open scientific work takes place on HPC resources, usually provided through the university and through the PIs of those universities. But in that way, we do have this open collaboration that lets us drive forward the science so quickly in collaboration with academia. So it's a very important partnership for us, although none of these are kind of commercial jobs. So we do the tool development in this open way, and then we apply the tools that we develop to our closed projects internally. All right, so I'll say a little bit about transformers because that's the hot topic of the year. Obviously, we heard about them already a little bit. They're used in many things such as machine translation. So, maybe you can view a transformer for machine translation, might translate from Swedish to English for my benefit because, my Swedish is not that great. But you could also imagine translating from one molecule to a better molecule. And that's kind of the paradigm that we have for the use of transformers internally. So we start a starting point and we can kind of make relatively small changes. We set these up in a way to make quite constrained changes. That improve molecules, but sort of borrowing from NLP and really well contextualized by what Joran said, we are also moving to a model where we can pre-train a transformer on an enormous amount of chemistry and then specialize it for sort of fine-tuning its performance. So exactly like natural language modeling. And actually, we have a collaboration with Nvidia. So right now, we're running experiments on the C1 computer of theirs, which is this large GPU cluster where we are doing sort of the largest ever chemical language modeling work. And so the rough idea is we take sort of the transformer model that we've built. NVIDIA has been helping us a lot really expand from what I would call a language model to trying to have something like a large language model. We're nowhere near the parameter space of NLP models really because the token set for chemistry is much smaller, but we're trying to get there. And then the idea is we can take really large chemical databases, make relatively large pre-trained transformers, and then we can adapt them for all of our downstream tasks. So that's another use in which we really are touching HPC responsibility. And again, this is all open and will be published. That's typically the model we have for this kind of collaboration. All right, I see I have an errant arrow. It's the danger of stealing people's slides. So this is one set of things and the things that I work on really, but there's another very important part of this, and this is that simulation aspect. So this also gets a lot into compute. What I showed you in the beginning was a static protein, and we were trying to fit molecules into that cavity, and we showed we could do that quite effectively. But proteins are not static, they're dynamic objects. And so in order to appreciate these dynamics, we actually need to simulate them. And so there's a technique called free energy perturbation. It's a type of molecular dynamics simulation. I won't get into the specifics of it here, but it's a computationally fairly expensive way. So I will say a molecular dynamics FEP-based assessment of one molecule against one protein takes about 5 GPU hours on a A100. It's not It's about how quickly we can go. It's not big enough to parallelize across multiple GPUs, but typically we want to do many molecules at once. So we do have a great need for many GPUs, but they all are running one molecule. But this is to show you, internally, we have done experiments. This is about 15,000 molecules that have been made and tested. This is their experimental affinity on the x-axis, and or maybe I have the wrong way around. This is the predicted, that's the experimental. And there's a correlation. It's not a perfect correlation, but if you consider how complicated biology is, for us, this is a really impressive result, and it's enough to justify investment in these methods. So FEP is now standard, and it's really a production tool in big pharma, not just at AstraZeneca and the whole industry. But we need to get faster than that. We need to get to larger idea sets. So we also have been working a lot with active learning FEP pipelines. So there we take our artificial or natural intelligence ideas. Everything just goes into the bucket. We assemble them into blocks, usually a couple of tens of thousands. Then we take some hundreds from that subset, we simulate them using FEP, we build a machine learning model to infer those results, and then we use a sort of active learning loop to pick more subsets to simulate. And these are kind of self-terminating processes that explore these blocks for promising molecules and then report back to us. These are the best results that we've found. And so when we combine these kind of active learning block workflows with our idea generators, like the molecules we get from reInvent, we have this kind of fully digital autonomous molecular design platform. So reInvent might generate hundreds of thousands of ideas, and then we take them in batches of 20 to 30,000 through the high throughput FEP system, and then we can really deliver to our experimental chemists shortlisted compounds that have, really good properties as we expect. But the implication of this is that each compound we make behind each chemical synthesis is actually potentially hundreds of GPU hours. And this number is only going to get more and more as we proceed. So this, I think, is also, an important part for this audience, as we've discussed a little bit about workflows. So in order to facilitate this high throughput simulation and the requirements of that, we have internally developed a workflow manager called iClass. And this is basically a wrapper around different types of molecular simulation. So we can front end into GROMACS, we can also front end into FEP, which is a commercial, Schrodinger's FEP module, which is a commercial alternative, and other things as well. And just to sort of give you a sense of how complicated these are, each one of these lines is basically a step in the simulation, or in some cases an entire simulation. So we need to kind of wrap all of these things. They need to happen in sequence, or in GROMACS or PMX in this case, in order to produce a result. And so we've done some benchmarking of these open source workflows compared to closed source alternatives. That's what this is, and we did this work in collaboration with one of the Max Planck Institutes. So this is Bert de Groot's group. And so essentially we have these, is also publicly available. We have this workflow manager that has a fully open source alternative to the closed source, but we can kind of toggle at the workflow level if we're running it open source or closed source, depending on our needs and the sort of token availability. So the licensing of the commercial software for these actually becomes quite limiting for us. And that's partly why we look into these open source alternatives. Okay. So that is the first two steps of a pipeline. So we have a system that can generate ideas, and hopefully I can motivate you that we can screen these ideas quite thoroughly using a lot of compute. There's a final step that we also work on, and that is synthesis planning. So once we have come up with our short list of ideas, we also use AI and computation to help chemists make these ideas. And so the way this works, we have a lot of data like this. So, you have this molecule and this molecule, you mix them together under these conditions and they react to form this product over here. And this data is available from the patent literature. There's around 2 million reactions that are patented that are available. And we also have our own internal records of reactions that have been done at AstraZeneca for 20 years. So we have quite a lot of data. And then you want to basically use these to predict how to make a new molecule. So if this is the molecule that the AI designs, we would like to say you disconnect it into these two pieces, and then also, recursively disconnect it until you have simpler building blocks that you can buy. So we have developed these models. They can either be template based. So in other words, we look at the literature and we basically cluster them into templates, or template free models, which are transformers. We actually use both. And then you have to do this kind of Monte Carlo tree search where you recursively decompose molecules. And each time you break a bond, you have a new set of reactants that you can look through. So we actually generate these synthesis trees, which we can propagate and come up with the shortest and cheapest routes. So these things connect actually to our vendor catalogs. So the systems actually know how much the things that they're suggesting cost. And they can say, this route is cheap and available from a provider. So that's the kind of information we can feed back with our web interface. So this is the kind of picture we give to a synthetic chemist, that we suggest making the molecule by combining these two pieces, and this piece you get by combining those two. And the sort of green effect there means actually those are in-stock reagents. So they could just go to our cupboard and pick that, and they would be sort of orange if they were purchasable. So we kind of give that information. And again, that's also all fully available and actually preloaded with the patent data. So you can download these as well. There's a little catalog of papers here from these group of people. So all of this is actually shared And this is a hot field, so there are a lot of people developing methods for retrosynthesis planning. There's commercial solutions, there's academic groups, and they kind of benchmark these tools together, and we kind of do all right in these competitions. But we work, our internal system has quite a lot more data, so we have some advantages there. Obviously, that data we can't share. All right, I think I have a little bit of a malfunction there with the middle box, but I will just quickly walk through this kind of conclusion slide. So the first thing I'll say is that, in the last maybe 5 years, I've only been industrial for 2 years, but my understanding is the extent of use of simulation has really exponentially increased over those 5 years. We've gone from MD being a kind of specialist function performed by a few computational chemists for special applications to a really common production system. And along with that, we have AI idea suggestion, which is now also widely applied. So we track impact, and we think about 68% of the projects at AstraZeneca are accelerated by these AI methods. So they actually have made compounds designed by AI. So we are no longer talking about AI designed chemistry, we're making it in labs. And this is only going to go, more and more and more toward the future. So I've already said we have a compute cost per molecule that we make, and that I think is only going to get higher and higher because the number of ideas that we explore for every idea we make in the lab is only going to get bigger. So we're going to have increasing demands for this kind of compute. But it is important to say that, especially with biological systems, the complexity of cells and organisms is so enormous that we have to also be humble that many of these things are outside of our current and even, medium-term predictive capacities. So protein ligand affinity is predictable, but toxicity is really, really difficult because if you can be toxic in so many different ways. A molecule can have so many off-target effects that, we'll never realistically be in a place where we're not going to have to do a lot of experimentation. And so that is important to say. And actually, we're looking at how we can kind of bring human in the loop modeling into these kind of generative loops. So get more human input as the runs proceed. And that's actually something that we're looking into in the future. We have a project with Alto around human interaction with these design agents. So that's all I had to say. Thank you for your attention, and any questions? More than welcome.